Hello and welcome back to Excelsior Academy. So in this session, we are going to actually summarize what we have already studied about the cyclins and cyclin dependent kinases. So this lecture is nothing but the summary of what we have already taken in the previous classes. So let's check out. Maybe 1 or 2 points will be extra. So it is actually easy way to understand and remember the cyclins. What are the different cyclins and cyclin dependent kinases? Okay. Yeah. So cell division cycle regulation. So all these regulatory subunits and all what we have studied in the previous lectures. So we know that it is crucial to ensure the proper segregation of genetic material for the normal development and maintenance of multicellular organisms. So this we know, and that's why the regulation of cell cycle is very, very important, right? Now, if suppose there is any failure, it will lead to obviously the cancer or the abnormal growth. So normally, whenever we say about abnormal growth, it's always meant that the cell has going to become the cancerous. Now, the key regulatory protein of cell division cycle is a heteromer, which consists of cyclin and cyclin-dependent kinases. So this already we know about this protein, and these are serine threonine protein kinases. Now, when you say this point is extra, that your CDK is actually a serine threonine protein kinases. So what does it mean? Serine threonine protein kinases. It means that cyclin dependent kinases will add phosphate group only to the serine or threonine amino acid of the substrate. Okay, so the protein or the substrate which is undergoing phosphorylation of that particular protein only to the serine or threonine, it will add the phosphate group since they have got the OH group present. So cyclin is the regulatory unit and CDK is the catalytic subunit. Okay. So CDK, they transfer the phosphate group. So as I already mentioned that in the kinases means that it is going to add the phosphate group. Now, phosphorylation causes change in the enzymatic activity of the substrate to interact with the other proteins. Now, from this, you need to understand that phosphorylation is changing the enzymatic activity, means that either it is going to activate the protein or it will also can make inactive the protein. So suppose, for example, we know that there is a regulatory protein V1, which is a kinases, which goes and phosphorylates different groups over there and makes it inactive, right? If you remember in the MPF regulation, if you remember the figure, you must have seen that in the MPF, that is maturation promoting factor regulation, V1 comes in and adds 3 phosphate group, which becomes inactive. Now later CDC25, which is actually a phosphatases, it will remove the phosphate group and makes it active. So from this, you need to understand that it is the phosphorylation or dephosphorylation which is actually regulating the enzymatic activity. So either it is going to make it active or it will go make it inactive. Okay. So now CDKs are activated by binding to its regulatory units called cyclins. Now हमें ये पता है कि हम अभी तक जो cell cycle में पढ़ते आए हैं that each phases of the cell cycle, whether it is G1 or S or G2 or M. So each phase has got its own particular cyclins. Okay. So 9 CDKs have been identified total, of which 5 are active in cell cycle. So during G1, CDK 4, 6 and 2. During S phase, it is 2, and during G2 and M, it is CDK 1. Okay. Now, this is also very important point to remember that cyclin CDK complex is formed as a result of interaction between the cyclin box, okay, which is a conserved region across the cyclins, and a region of CDK which is called P stere. So what you need to remember is that whenever we show that cyclin and CDK is binding, so this which is present in the cyclin, it is called the cyclin box, and the region which is present, which is interacting with the cyclin, it is called your P stere, which is present in the CDK. Okay. Now, this is one example shown over here. This is the example of CDK2 interacting with cyclin A. Now, you can see that in the figure also, sorry, you can see that over here, the CDK2 is having the ATP binding pocket. Now, why this is required? Because you know that CDK2 is a kinase, and kinase is going to add phosphate group from ATP. Okay, so this phosphate group, that is from the ATP hydrolysis, you get a phosphate group, and that phosphate group is added to the substrate. Okay, now if suppose somebody asks you like from where the kinases is getting their phosphate group, then you should understand that they get their phosphate group from the ATP. So it means that CDK2 is also having a ATP binding pocket. Now if you see in the figure, see the cyclin box is there, which is actually interacting over here. It is interacting with the p-stere helix of the CDK2. So these are the helix over here which is present, and it is the p-stere helix and the box which interacts. So two side views has been shown interacting the CDK2 and the cyclin A. Okay. Okay, so this is the cell cycle cyclins. So from here, it is very much understood, and this all, I mean, many a times this will be a repetition also whenever we talk about cell cycle. So you can see that whenever G1 phase is there, okay, so you can see that what kind of cyclins are getting activated is a G1S cyclin. So even though in few textbook it is mentioning as G1S cyclin, and some cases it will be mentioned as cyclin D. So you should know that it is the cyclin D which gets activated or which goes and interact with the CDK4 and 6. Okay, so we know that in G1S or G1 phase, we have got the cyclin D, which actually interacts with your CDK4 or 6. Sorry. Okay. Similarly, when it enters the S phase, you have got the SCDK and S cyclin. So here it is cyclin E and CDK2. Now, in the case of G2 or M cyclin, it is your CDK1 and the cyclins are cyclin A and B. Okay. So, either you can remember in this way or you can remember in the alphabetical order also. Now, let's see the graph over here. So, what does the graph says here? See, you can see that in the G1 phase, that is the start point. Remember the start point, or which is also called the restriction point, right? So if it is crossed, then it means that the cell is fated to enter the S phase. Okay. Now, so this is very important and crucial. So you can see that over here, the cyclins, the concentration of G1S cyclin is increasing and it starts decreasing towards the end of the G1 phase, or you can say the early phase of the S phase, right? Now, one more thing is important to understand is that the moment the cell has crossed the start point, okay, or the restriction point, the moment it has crossed the restriction point, the synthesis or the increase in the concentration of S cyclins, for example, your cyclin E, okay, the concentration starts increasing at the moment the cell has crossed the restriction point, because you know that crossing the restriction point means the cell is fated to enter the S phase. Now, even after that, if suppose there is a damage to the DNA, still the cell is going to enter the S phase. Okay, so the moment it has been cross, the concentration of the S cyclin or cyclin E starts increasing. Okay, and it will be throughout the, you can see that it will be throughout present and it will get over at the end of the G2 phase. Okay, now the moment the cell has crossed the S phase, that is your DNA duplication or replication has been completed. Now, the cell enters the G2. Now, the moment S phase has been completed, it will enter the G2 phase. Now, where G2 will ensure that all the machinery or the requirements are ready so that the cell can enter the M phase, correct? So, at that point, that is towards the end of the S phase or the starts of the G2 phase, your M cyclin or the cyclin A and B will start increasing. Okay, so now it is going on increasing, and towards the end, where the metaphase anaphase, where the metaphase anaphase stage is there, it will start decreasing. So this is how you know that the cyclins are increasing and decreasing and regulating the cell cycle, and that's why these proteins are called cyclins, because They are fluctuating like a cycle, going up and down, going up and down. Now we know that at the end of this, again G1 is there. So G1 phase is coming. So it means what? Again, the G1 S cyclin will start increasing. And this keeps on continuing till the cell is under normal control of the cell cycle regulations. If suppose something goes wrong, then either it is meant for the apoptosis or it will become the cancerous. Okay. So I hope this is clear. Now the same thing has been explained in the different diagram also. So here also it is the same thing with the different terminology, but the same concept over here. So you can see that in the G0 phase, you know that it is also called the resting phase or the quiescent phase, where the cell is inactive and it is not undergoing any phases of the cell cycle. Now, suppose it enters the G1. It has seen that all the growth factors and the nutrients are available. So the G1 phase has been started. Now, with the help of the cyclin D interacting with the CDK4 and 6, which is present on your G1 phase, it is crossing your restriction point. Now, once the restriction point has been crossed over here, you can see that the concentration of cyclin E and CDK2 will start increasing. Okay. Now, this will keep on increasing till your S phase has been completed. Now, for the S to continue, the E cyclin has been replaced by the cyclin A. And then it is entering the G2 and M phase where both cyclin A and B is helping. So, this is how different stages of the cell cycle has got the different cyclin dependent kinases and the cyclins. Okay. So, we'll just study about the cyclins in detail again. So we know that there are different cyclins which is required at different stages of the cell cycle. So already mentioned that the CDK or the cyclin CDK complex, we know that the D1, D2, D3 binds with 4 and 6 and it is necessary for G1. Where E binds to CDK2, it is required for the progression from G1 to S. And in the S phase, you have got the cyclin A binding to CDK2 complex. So now from this, you should understand that whenever CDK2 is present with A or E, it will tell you what stage the cell is present. Suppose a question comes that you have found the complex of CDK2 in association with cyclin A. So the student should understand that the cell is present in the S phase. If suppose it is found with cyclin E, then you can say that the cell is progressing from phase G1 phase to S phase. Okay, so the amount of the complex found can also determine the stage or the phases of the cell cycle. Now, in late G2 and early M, cyclin CDK1 promotes the entry into the M phase and mitosis is further regulated by cyclin B CDK1. Okay. So the cyclins, more information about the cyclins. So they are named because the concentration varies in cyclical fashion during the cell cycle. Obviously, since it's a cycle, so as I already mentioned, the cyclins are also proteins. So their concentration is increasing and decreasing as it is progressing from one phase to the another phase. The oscillation of the cyclins, namely fluctuations in cyclin gene expression and destruction by the ubiquitin-mediated proteasome pathway, induce oscillations in CDK activity to drug the cell cycle. So we know that there is a oscillations of the cyclin, that is the expression and destruction of the cyclin is happening. So in a way, it is eventually affecting the activity of the CDK also in order to regulate the cell cycle. A cyclin forms a complex with CDK and it begins to activate, but the complete activation requires phosphorylation as we have already mentioned, right? So the complex formation results in the activation of CDK active site, and finally active means what? Then it can add the phosphate group to the substrate. So cyclins themselves have no enzymatic activity but have the binding site for some substrate and the target. So the CDK to specific subcellular locations. Now different types of cyclins. So already we have talked about different types of cyclins like cyclin D, A, E, B are present. So just more information on these cyclins. So G1 or S cyclins rise in late G1 and fall in the early S phase. CDK G1 cyclin complex begins to induce the initial process of DNA replication by by arresting system that prevent the S phase CDK activity in the G1. Okay. So if you remember when we were talking about the S phase and the DNA replication, so it was the CDK4 and 6 which was helping in the initiation of the DNA replication, if you remember the figure. If not, please go back and watch the video again. The cyclists also promote their other activities to progress the cell cycle, such as centrosome duplication and the spindle body formation. Now the rise in presence of G1 S cyclin is paralleled by rise in S cyclins. If you see the figure over there, you can see that as the G1 is increasing, you can also see that the S cyclin is also increasing. Yeah. Now, G1 cyclins do not behave like other cyclins in that the concentration increase gradually throughout the cell cycle based on the cell growth and the external growth regulatory signals. Okay. So, the presence of G cyclins coordinates cell growth with the entry to a new cell cycle. So, what does it mean? It means that if suppose there are no external growth regulatory signals, there won't be any synthesis of G1 cyclins. Eventually, the cell will get arrested at the G1 phase and it will not progress to the S phase. It means what the cell has been arrested. Okay. Now, Kaposi sarcoma herpes virus encodes D type cyclin. So, this will go and bind to CDK6 and it will contribute to the cancer. So, Kaposi sarcoma is actually a type of cancer. So now we know that it is encoding the D-type cyclin and regulating the cell cycle differently by binding to CDK6. So this is how cyclin D is regulating. I'm sorry, I couldn't type it everything, but still it is given very nicely and it is readable also. So cyclin D synthesis is initiated during G1 and drives G1 S phase transition. So when growth factors and mitogens or nutrients are present, the cell will start expressing the cyclin D or otherwise called G1 or S cyclins. Now growth factors are there, so it will work through the RTK pathways. So this is something about the cell signaling. So it will work through the RTK pathways and stimulate the RAS, RAF, ERK kinase pathway. Now this RAS pathway will lead to the activation of MIC. So this MIC will increase the rate of transcription of cyclin D and hence cyclin D will initiate the cell cycle and interacts with the two CDKs, that is 4 and 6. So what is happening? So you know that in the surrounding of the cell growth factors are present. Okay. So you see the growth factors are there. Now this growth factors will initiate your RTK pathways, which will stimulate your RAS pathway. And now this will increase the concentration of MIC, which is a transcription factor. And this will also increase the transcription of cyclin D. Okay. So you see that till now we all were talking about cyclin D and it's interacting with CDK4 and 6 and how it is, helping in the G1 phase and S1 phase. Now how cyclin D, like who and what initiates the synthesis of cyclin D? It's a protein. So protein means it needs a gene expression. Now how it has been regulated? This is the way it is regulated. So you have a growth factor which works through the RTK pathways and stimulates the RAS pathway, and it will lead to the activation of the MIC, which will increase the rate of cyclin D transcription. Okay. Now, similarly, the CL13 is a yeast homologue of cyclin D. It interacts with CDC28, that is your cell division control protein, during G1. Now, in proliferating cells, because of continuous signaling by growth factor, the cyclin CDK4/6 cyclin D complex starts getting accumulated. Now, this is of great importance for the cell cycle progression. Cyclin D CDK4/6 complex partially phosphorylates RB tumor suppressor protein. So remember retinoblastoma, this also we have talked about how it is helping regulating the cell cycle. So once this phosphorylation of RB is important for S phase progression. The phosphorylation of RB causes dissociation of RB from the E2F transcription factor of the cyclin E. So then what will happen? Since this complex is getting activated, cyclin D CDK4, this goes and phosphorylates RB. So now RB is not able to bound to the E2F, which is a transcription factor for cyclin E. So now your E2F is a transcription factor which is free of RB. Okay. So now your cyclin E is free, or it is actually transcribed for the S phase progression. So now are you getting how actually cyclin D is working? See, so cyclin D is there. It interacts with your CDK4 and 6. Now this gets activated and gets RB phosphorylated. Okay. So it is going to phosphorylate your RB. Now since it is phosphorylated, it is no longer bound to E2F, which is a transcription factor for cyclin E. So since RB is phosphorylated, E2F is not able to bound to RB. E2F is free. It is free. It means what? Cyclin E has been transcribed. And it means what? If there is an increased concentration of cyclin E, now the cell has entered the S phase. Okay. So read more and more and try to write in the flow chart so that you can understand what exactly is happening in the cell cycle. because each step, based on these steps only, the questions will come in the exams. So now coming to the S cyclins or cyclin E. So they bind to the CDK2, that is over here, and induces the DNA replication. So the levels of S cyclins remain high not only throughout the S phase but through G2 also and early mitosis, as well as to promote the early events in the mitosis. Okay. So we know the cyclin E is there, which goes and binds to the CDK2. Now, this complex of cyclin E and CDK2, it phosphorylates your P27 kip. Now, what is P27 kip? It's an inhibitor of cyclin D. Okay. So cyclin D remains unbounded form as the cyclin E is already bounded with the CDK2. The phosphorylated form of P27 is tagged by ubiquitin and get degraded by the UPS. That is your ubiquitin proteasomal complex. Thus, promoting the expression of A, allowing progression to the S phase. Okay. So this is how E cyclins are regulated. Now coming to the M cyclin. So M cyclin means here we are going to talk about the cyclin B. So M cyclin concentration rise as the cell begins to enter mitosis and the concentration peaks at the metaphase. So remember, till metaphase, anaphase, this M cyclin will be there, and after that it will drop. Cell changes in the cell cycle like the assembly of mitotic spindles and alignment of sister chromatids along the spindles are induced by M cyclin CDK complexes. So remember when we talked about the M cyclin CDK complex, it helps in the chromatin condensation, it helps in the nuclear envelope defragmentation, it also helps in the spindle fibers formation. Right. So the restriction of M cyclins during metaphase anaphase after the spindle assembly checkpoint is satisfied, it causes the exit of the mitosis and cytokinesis. So you see that the moment the cell has crossed the metaphase anaphase stage, the level or the concentration of M cyclins decreases. It means that now the cell is going to exit the mitosis and the cell is going to divide into two daughter identical cells now. So the expression of these cyclins can be detected immunocytochemically, that is, with the help of the labeled antibodies in individual cells in relation to the cellular DNA content. So you can also study about the initiation and termination of the DNA replication during S phase, and everything can be measured by the flow cytometry or otherwise called FACS. So cyclin A and B. Cyclin A plays important role in the regulation of two different stages of cell cycle. Association of A with CDK2, it means it is in the S phase. If it is associated with CDK1, then it means it is required to enter into the M phase. Okay. Now, B is a mitotic cyclin only. You can see that A is present for both S phase as well as for the M phase, right? Whereas B is only meant for the mitotic cyclin. So, the complex of CDK and cyclin B is called maturation promoting factor or mitosis promoting factor. Cyclin B is necessary for the progression of the cells into and out of the M phase of the cell cycle. So, this already we have studied in the previous lecture also. So cyclin B synthesis starts in the S phase only. And of course, why? Because all these protein synthesis will only happen in the S phase only. Because all the proteins which is required for the cell cycle gets actually accumulated before itself. And then it is exported out of the nucleus because it has nucleus exporting sequence. So NES. Cyclin B and CDK1 complex activation causes breakdown of the nuclear envelope. And finally, it enters the prophase of the mitosis. Now, if it gets deactivated, it means it is going to exit the mitosis. So, during the late anaphase stage, cyclin B concentration falls due to degradation by the UPS. So, finally, what is happening now? The cell is exiting the mitosis. So, this was all about the different types of cyclins which are present in the your cell cycle. So, this is the analogy given over here, the various levels of CDK and the cycle during the phases of the cell cycles. You can see the G1 is there. So you have the moment the S phase it enters, H phase CDK will come. Then at the initial stage of G2 itself, mitotic CDK will come, right? So this is how your CDKs are activating. Now, whereas about the cyclins, you can see in the G1, you have got your D1 cyclins, D2 cyclins are there, right? The moment it is entering the S, you have got the A. So different cyclins are also there. So you can compare and you can study about the relative amounts of these cyclins also. Okay. So cyclin A and B, they have got the destruction box, and cyclin D and E contains the pest box. Now what is pest? It stands for the proline, glutamic acid, serine and the threonine. So these are required for efficient ubiquitin mediated cyclin proteolysis at the end of the phase of the cell cycle. So what does it mean? It means that, see, now till now we were talking about how these cyclins have been increasing the concentration. Now they need to be decreased or they need to be restricted. So how? So A and B have got their destruction bonds, whereas D and E contain these sites or the Pest sequence to which ubiquitin will be added so that it will be targeted for the proteasomal degradation. Okay. So ubiquitin mediated proteolysis of cyclin is catalyzed by ubiquitin ligase enzyme. So SKP1 colon F box and the anaphase promoting factor complex is responsible for the ubiquitination and the destruction of the cyclins. Please remember, this is very important with the in view of the exam, especially the MCQ. And now talking about the regulation of CDK. So already this has been taken in the previous lectures with the beautiful diagrams. So this is just the summary of how CDK is helping in the regulating the cell cycle. So there are three mechanisms, either undergoing the synthesis and degradation of the cyclins, or you can do the phosphorylation, or you can bind with the inhibitors. Now what happens when you actually phosphorylate? So this is just now, earlier also I was discussing. So you have got the V1 and MIT1 protein kinase which will phosphorylate the CDK1 at three different sites. So here we have mentioned about the tyrosine 15 and 14. So remember, there is one more is there. I think 161 tyrosine is there. I think it is 161. So phosphorylation will inhibit the activity of CDK1, then CDC25 will come and dephosphorylate, making it active. So dephosphorylation at these site is necessary and CDC25 phosphatase does it, right? Now full activation of the cyclin CDK complex occurs when CDK7 and H complex, okay? This is something new we are studying. It will phosphorylate threonine 161. Okay, sorry, it is not tyrosine, it is threonine only. Okay. So near the CDK active site. So this is something new we are learning now. So how it is? So we know that tyrosine 15 and 14 gets phosphorylate, and it also phosphorylates your threonine 161. Now this is done by the activity of CDK7 and cyclin H. Now when CDC25 comes and removes this phosphate, then only your CDK gets activated. Okay, the other method is by binding with the inhibitor. So that is CKI, that is cyclin dependent kinases inhibitors. So CDK activity can also be regulated by cell cycle inhibitory proteinase, that is CDK inhibitors, which will bind to CDK alone only or to the complex also of the CDK and cyclin. So there are two families are there, INK family and CYPK family. This also we have discussed. So INK family will inactivate G1 CDK, and CYPK family will inhibit the CDK2 activity. So both are internal and external signals are responsible for the regulation of CKI. So this is how the CDK activities are regulated for the maintenance and the regulation of the cell cycle. So that's all for today. I hope everything was clear, and this lecture is very, very important for you guys. So please study well, and if any doubts are there, please ask in the comment section, or you can also mail to us at excelsioracademy1228@gmail.com. You can also ask your queries in the Telegram channel, and you can search the Telegram channel with the same Excelsior Academy. So you can see over there also, you can ask, join us and ask your queries. So I hope you understood the today's topic, and soon we are going to complete the cell cycle. Many more few questions are also there, and I'll be taking few experiment based examples also, so that you can understand. So whenever a researcher or scientist does the experiment, how the experimenter or the researcher is actually interpreting, and how exactly you understand what kind of work is going on. Okay, so all these lectures are going to help you in preparing for your future exams, as well as also to understand how the research is being carried out in the most of the labs. So thank you, and keep watching. If you like it, please share and subscribe. Please share with your circle, friend circle also. Thank you.